So my talk today is about Ym1 and Ym2 and how they regulate communication between the immune system and the extracellular matrix. As Andrew said, I'm a Wellcome Trust Immunomatrix PhD student supervised by Judy Allen, Tony Day and Pedro Popotto. And this project is in collaboration with PhD student in Tony's lab, Catherine Malpass. So today I'll be presenting work from both of our PhDs. So the chitinase-like proteins. So the human CLP, or chitinase-like protein, YKL40, is a biomarker for poor prognosis in a number of pathological conditions, including cancers and fibrotic diseases, including COVID-19, and is associated with poor lung function in asthma and COPD. And YKL40 is thought to be functionally equivalent to Ym1. And Ym1 and Ym2 are the mouse chitinase-like proteins. And all CLPs are closely related to enzymatically active chitinases, molecules that break down chitin, but they lack this enzymatic ability due to loss of function mutations. However, it is known that YKL40 can still bind to chitin, as illustrated here in this chitin binding groove. Ym1 and the closely related Ym2 are both markers of type 2 immunity. However, despite this knowledge, exact sort of mechanisms of action and functions of these proteins remain unknown. So what do we know about the function of Ym1? So firstly, Ym1 has been implicated in eosinophil chemotaxis, although this is somewhat controversial. And previous work in our lab has outlined a role for Ym1 in the regulation of IL-17. Ym1 is also known, it's been known for a long time, that Ym1 binds heparin, which is a GAG or a glycosaminoglycan. And GAGs are long unbranched polysaccharides and key components of the extracellular matrix. We hypothesise that Ym1 can also bind heparin sulfate, although this hasn't been shown. We think that Ym1 will bind these GAGs within this chitin binding groove, similarly to the YKL40 on the previous slide. But what we don't know is the role that this GAG binding plays in the underlying mechanisms of action of the CLPs. And first of all, I'm going to talk about the work we've done to unravel this mechanism of IL-17 regulation. So what are the molecular requirements for IL-17 induction in gamma delta T cells by Ym1? So IL-1 beta and IL-23 are innate cytokines, which are potent inducers of IL-17 within gamma delta T cells. And previously it was thought that Ym1 acted directly on IL-1 directly to induce IL-1 beta and act on gamma delta T cells to release IL-17. But an alternative explanation is that Ym1 is acting in synergy with IL-1 on these gamma deltas. So to look into this, we did 3 in vitro assays. We took peritoneal exudate cells, or PEC, from black 6 mice and incubated these cells for 48 hours with IL-1 beta, IL-23 stimulation alone, or the cytokines alongside 2 concentrations of Ym1. And what we found was an increased frequency of the total IL-17 positive gamma delta T cells. And gamma delta T cells can be broadly subcategorized into 2 subsets, those that produce IL-17 and those that produce interferon gamma. But the plasticity of these cells can give rise to a double positive population, which express both IL-17 and interferon gamma. And when we look more closely at this increase, it was clearest in this double producing population. However, this is looking at whole PEC, and it is still possible that accessory cells within the sort of peritoneal exudate cells could be releasing IL-1. So at this stage, we still weren't sure whether it was Ym1 was acting to induce or synergize with IL-1. So we asked ourselves a question, is Ym1 acting directly on the gamma delta T cells? And to do this, we repeated our experimental setup, but this time using FACS-sorted gamma deltas. We again incubated for 48 hours with the cytokines alone or in addition with Ym1. And again, we saw this increase in the double positive population illustrated here and also in the FACS plot. So this is with the stimulation alone and this is with the addition of Ym1. So this confirmed to us that Ym1 was acting in synergy with IL-1 to upregulate IL-17. So we know that Ym1 is acting directly on these gamma delta T cells, but we don't know exactly how. So one hypothesis is that it's acting directly via a cell surface receptor, for example CD44, which we know is upregulated on gamma deltas. Another hypothesis is that Ym1 is interfering potentially with the glycocalyx via GAG binding and exposing the IL-1 receptor. But what happens with Ym1 and IL-17 in vivo? So to look into this, we took male black 6 mice, divided them into 4 groups. On day 0, we gave an intraperitoneal injection, and on day 2, we collected the PEC. The 4 groups consisted of PBS injected mice, and those injected with Ym1 alone, LPS alone, or Ym1 and LPS. We chose LPS because we know that this induces the production of IL-1 beta and IL-23 from macrophages and monocytes, amongst other things. And what we saw was that when LPS was administered to these mice, we saw a significant increase in the IL-17 positive gamma delta T cells. But when Ym1 was administered with LPS together, over here, you can see that this response is limited. So we asked ourselves, is Ym1 interfering with the action of LPS? And if so, how? So to look into this, we repeated the in vivo setup, but instead of having a Ym1 alone group, we had a group of mice that received LPS initially and Ym1 3 hours later. And interestingly, we saw with LPS, we saw the expected inflammatory response. So we saw the neutrophilia and an increase in Ly6C high monocytes. However, when LPS and Ym1 were given together in the turquoise bars, you can see that the response in terms of these cell populations is the same as the mice that received PBS. However, when LPS and Ym1 were administered separately, we again saw this inflammatory response. So therefore, we hypothesized that Ym1 is interfering with the action of LPS by direct binding, which ultimately reduces the amount of pro-inflammatory cytokines. But what is the importance of GAG binding for these CLP functions? So an important tool that's been developed by Catherine is recombinant Ym1 and Ym2. And these recombinant proteins have enabled us to better define the GAG binding properties of the proteins. And just a reminder that a GAG is a long polysaccharide, a glycosaminoglycan, and a key extracellular matrix component. And we hypothesized that this is the GAG binding site. So first of all, the binding of Ym1 to heparin was investigated, and heparin of varying chain lengths was used. And as you can see, as the heparin chain length increased, the strength of the binding also increased, illustrated by a low KD. Catherine also demonstrated the binding of Ym1 to heparin sulfate, which hadn't previously been shown. We also looked at the binding of Ym1 to chondroitin sulfate, keratin sulfate, and dermatin sulfate, as well as hyaluronan, and showed that Ym1 does not bind to these GAGs. Taking a closer look at the structure of heparin, it's possible for heparin to be sulfated at 3 different locations, the 2O, the 6O, and the N-sulfation, and we can modify the heparin disaccharide in the lab to test varying sulfation patterns, and that's what we did. So we looked at unfractionated heparin, which has all the sulfations, and selectively desulfated heparins with a sulfate removed at each one of these points, and then fully desulfated heparin. And what we can conclude from this is that the sulfation at the N-sulfate is key for binding, because as you can see, when that one was removed, there was no longer binding. Ym1 and Ym2 proteins are known to be highly identical, about 94% by sequence alignment. However, we know that they're expressed by different cell types and upregulated in different tissues, as illustrated by this Hyperion lung image, where you can see Ym2 positive cells lining the airways and the Ym2 positive cells scattered within the parenchyma. Despite this high similarity between the 2 proteins, there are clear differences in the strength of heparin binding, as you can see on this graph, where Ym1 has much higher binding than Ym2 at every pH, as illustrated by requiring a greater salt strength to elute the protein. Both proteins, interestingly, exhibit a strong pH dependency, with both of them having a stronger binding at acidic pH. And this has potential implications for pathological conditions, as in certain inflammatory settings, for example, hypoxia, we know that the pH will be reduced, and this could then regulate the GAG binding. Another important tool that's been developed are mutant recombinant Ym1 and Ym2, and these have been designed, expressed and purified in the same way as the wild-type proteins, And with these mutants, the positively charged lysine residues have been mutated either to alanines with neutral charge or glutamic acid with a negative charge. And the aim of this is to reduce the GAG binding capacity. And preliminary data suggests that this has worked well. As you can see here with both Ym1 and Ym2, the mutant proteins do exhibit weaker binding. So to summarise, we have enhanced our understanding of the function of Ym1. We've hypothesised that Ym1 acts directly to bind LPS. We have also shown that Ym1 acts directly on gamma delta T cells to regulate IL-17. And we have also defined the GAG binding properties, showing a pH dependency for both Ym1 and Ym2 and the importance of the N-sulfation of heparin. In the future, we want to use the mutant recombinants, both in vitro and in vivo, in these systems to assess the importance of the GAG binding. And we'd also like to utilise the Ym1 knockout mice that are now available, and hopefully we can use the wild type and the mutant recombinants in rescue experiments. I'd just like to acknowledge all members of the Alan and Day labs, and particularly Pedro for his supervision, and Catherine for her biochemistry work, as well as the work of master's student Hannah Murphy, who did a lot of the immunology alongside me. I'd also like to thank groups that work closely with Judy and all of the hard work of everyone in the BSF and the core facilities. Thank you.
Okay, thanks very much, Georgia. That was really clear and nice. Does anybody have any questions for Georgia? Mark.
Hi, Georgia. Really nice talk. So in one of the models, you had the Ym1 acting on potentially some different receptors.
Yeah.
Have you any idea how they could potentially be working through those receptors? Do you think they're changing their conformation and their ability to bind to ligands? Do you think they're, directly signalling through that receptor? Have you got any kind of models?
Yeah, so at the moment, I think it's very early stages, and I think that they're sort of our next steps and what we really want to get to the bottom of. So we hope to utilise some cell lines where we can work without CD44, basically, and also cell lines where we can manipulate the glycocalyx as well, to see how that might be happening.
Time for one other question. I think Tracy's got one, unless somebody else does.
I noticed you were very clear to point out you were using male mice. What happens if you use female mice?
So that's a good question. We haven't done anything with female mice at the moment, so yeah, it's an interesting thing to check.
Any other questions? If not, I've got a quick. So you mentioned about and you showed us actually how different constituents of the whatever it is that Ym1 binds to can affect what it's doing. How much is known so far about how complicated those sort of complexes of Ym1 are, and is it different in different disease settings?
So I think it's definitely different in different settings and in different tissues, but I think it's still, again, early stages. We don't know exactly the proportions of the different GAGs that are present, and obviously we showed how pH can affect things as well. So I think it is very complicated. Hopefully we'll be able to unravel it more as we go.
Okay, if there's no more questions, thanks again, Georgia.
Thank you.